very much. So as mentioned, today we're going to be talking about using these artificial gut systems to protect us from gut infections and building into this idea of antimicrobial resistance and how we start to use some of these tools and ideas that we're going to be talking about here to really address some of these issues. So the gut microbiota, what do I mean by that? Well, I'm referring to the diverse and complex microbial community that actually lives inside each and every one of your guts. So this actually is quite variable in the different segments of your gut. So in your small intestine, you usually have sort of a more simplified or lower density of bacterial cells that live in there. As you move on to your proximal colon, you start to get higher concentration of nutrients and therefore more active microbial growth. And transverse colon, we start to get more bacteria growing here. We're getting a depletion of nutrients, which is going to affect the bacteria that like to live there and change the pH. Then as we move through your distal colon, again, we're going to have a lower concentration of nutrients, so more fermentation of protein, and again, a different composition of bacteria. So it's not only different in each of the different intestinal segments, but actually in each and every one of your guts. You all have a unique gut microbiota, so you can think of it as being unique to each of you like a fingerprint would be. So, and there are a variety of different ways that the gut microbiota can act to protect our health as well. So let's go through a bit of a gut microbiome in numbers for some of you who may be sort of new to this idea of a gut microbiota. In general, you have a microbiota not only in your gut, but also in other places of your body. For example, the skin also has a microbiota, and the mouth has a microbiota. So including all of the microbiotas that we have in our body, you have about 100 trillion microbes that live both in and on your body. And if you think about how many human cells we have and how many microbial cells we have, you actually have slightly more microbial cells in your body than human cells. So there's about 1.3 times as many microbial cells than human cells. If we start to look at the number of genes we have, actually bacteria have a wide diversity of genes. So about 150 times more microbial genes than you have human genes. And about 95% of that microbial diversity you have in your whole body actually resides in your gastrointestinal tract, your gut. I mentioned previously, each of you has a unique gut microbiota. And it's really important to start to think about the microbiota in the context of health, because it's thought that in some way, about 90% of diseases can be linked in some way to the health of the gut and the microbiome. And one other thing to think about with the microbiota is that it's actually more accessible and easier to manipulate than the human genome. So there's really potential to use the microbiota to promote health or to treat disease. So we're going to hear me talk a bit about this idea of colonization resistance in this talk. And what do I mean by colonization resistance? So what I'm referring to is all the different ways that your gut microbiota can help to prevent the colonization or resist colonization by pathogens, so bacteria that will cause disease. One method you might have heard of before is called through these molecules or chemicals produced by bacteria called short-chain fatty acids. And these are metabolites that these bacteria produce by the fermentation of things like fiber. Another molecule or metabolite you might have heard before are bile acids, and the gut microbiota is actually responsible for the metabolism of these bile acids. And depending on the different bile acid pool you have in your gut, this can in some ways protect against or even promote the growth of intestinal pathogens. Another thing to think about with your gut microbiota is the nutrients. So there's only a limited amount of nutrients you have in your gut, and if you have a healthy microbiota, it's possible that these microbes are eating up all these nutrients or acting as their food source, and therefore the pathogens wouldn't have food available for them to grow. But if you have a compromised microbiota, for example, if you've taken some antibiotics, you might kill some of these gut microbes that normally would use those nutrients and therefore could promote the growth of pathogens by providing it with the food source. There's also these molecules called bacteriocins. These are just small molecules produced by members of the gut microbiota or another bacteria that can, in some ways, inhibit the growth of pathogens, but while allowing the actual bacteria producing them themselves not to be resistant. So they're producing these molecules that are going to target bacteria, but not target themselves. There's also this idea of bacteriophage. So actually, I didn't mention it earlier, but you have a lot of viruses that live in your gut as well. There's about 5 times as many viruses as there are bacterial cells even. So some of these viruses actually target other bacteria and can kill bacteria. So lots of groups are looking at some of these bacteriophages, which is what these viruses are called, to see if they can treat specific infections by targeting these pathogens. And finally, along the gut, you also have a layer of mucus that sort of acts as a barrier to protect you from, sort of separate your gut itself from the bacteria that live in your gut. And if that barrier or mucus layer becomes compromised, then potentially some pathogens could then start to infect your gut. So if we can maintain that mucus barrier, it's also another way to protect your gut and your health. I mentioned earlier this idea that your gut microbiota can be protective and it can be compromised when you're taking things such as antibiotics that can kill some of these beneficial bacteria. But the idea then came about, if your gut microbiota is protective and sometimes you lose some of those protective bacteria, what if we replace those protective bacteria in the form of what's called a fecal transplant? And it is what you think it is. So basically, you take stool from a healthy donor who hasn't taken antibiotics or doesn't have any disease, and you homogenize it, and you load it into a syringe, or some cases it's loaded into a pill, and you administer it to a patient. So either one of two routes. So you can give it down a tube that either goes into your stomach or the beginning of your small intestine, or you can deliver it as an enema or as a colonoscopy. So delivering it in those two different ways. And this fecal transplant, or FMT as I'll call it, is one of the first generation microbiome therapies that we have. So the idea, as I mentioned, you have someone who's got a disease state and you're trying to replace the gut microbial ecosystem through an FMT, restore them to a healthy state. As you might imagine, there are several disadvantages to this. The first being everybody has such a diverse and complex gut microbiota. We can never be 100% sure what is in that mixture. There is even some bacteria that colonize the gut that we've not even named yet. They're sort of unknown bacteria. So we don't have, and also because everybody is so individual, so because everyone's gut microbiota is unique to them, it can be difficult to know exactly what we're administering to people. There's also the risk that we potentially transmit infections, which could be a particular problem for people who are immunocompromised. So we do our best to screen stool from our healthy donors, but there's either the possibility we're not screening for pathogens that we should be screening for. There's been the case, I believe about a year ago in the United States, where a patient actually passed away because they had received an FMT from a donor where they're not screening for a antibiotic-resistant bacteria. So there's also been some other cases where patients did receive infections, even though their stool samples were screened, but their screening method wasn't sensitive enough. So that's always a possibility with FMT, no matter how careful we are. Another thing we need to think about is whether the bacteria from this FMT can engraft. What I mean by that is, can they actually colonize and live in the gut? Because you do have some bacteria that are there originally, so they might be competing for the bacteria in the FMT treatment. So there's no point in administering this FMT if they're not going to end up colonizing and living in the gut. Another thing is to think about what patients we're giving this to, because some of these patients are going to go on to receive lots of antibiotic treatment for other conditions or because they're immunocompromised. So if we're going to be giving them an FMT treatment and then we know within a week they're probably going to need chronic antibiotics, there's no point to really administering the FMT in the first place, because you're essentially undoing the purpose of the FMT procedure. And finally, we don't really have a good understanding of the long-term effects of FMT. It's a relatively new procedure. It's really started to be used more popularly since from about 2013. So we know we're transferring this complex community of bacteria, and increasingly we're seeing all these different studies that are linking the composition of the gut microbiome to a variety of different disease states. So we really need to be sure that we're not going to potentially transfer bacteria that can lead to later disease, such as colorectal cancer, for example, or diabetes. And finally, we're seeing that some FMT donors can be better than others. There's been this term out there called super donors. And these are donors that just seem to work really well in some patients, but they can be difficult to identify in the first place. We don't really know until we've used them in the patient. And once you have that donor, it can be difficult to retain them. For example, we need to make sure that that donor doesn't go on to receive antibiotics. We can obviously control that all the time, but it could compromise their gut microbiota and might make them less effective. Because of this these challenges, we've developed the idea of second generation microbiome therapies. So the idea here is that instead of giving the entire FMP or the entire stool donation from a healthy patient into a diseased patient, what we do now is to design what's called a synthetic cocktail of bacteria. So this is where we culture bacteria from the stool sample, and we sort of mix them together in a defined mixture, so we know we're giving them, and we then administer that to the patient. So it's a more controlled way you can think of, or a more clean FMP. And while because it's defined, it can be safer than an FMP treatment, it's still not exactly perfect, because there's still a lot of questions we need to ask, such as what bacteria we want to include. So we need to have an idea of what these bacteria are doing in the gut and what their mechanism is before we can start to decide which bacteria are going to be in the synthetic cocktail. How safe are the bacteria? So they need to be tested under a variety of conditions, and how bacteria might behave in the lab when grown on its own is not necessarily how it might behave when it's grown with other bacteria that are in the gut. So it really needs to undergo a lot of testing. And again, we need to make sure that it's going to be able to colonize or live in the gut as well, and it's not just going to be washed through. And this is a problem that you might have heard when we're talking about traditional probiotics treatments. So often, in a lot of cases, these probiotic treatments need to be taken continuously because they're not grafting and they're not living in the gut. Again, we need to make sure that these patients don't go on to receive a lot of antibiotics. So if they do need to be given the synthetic cocktail, it's probably a lot easier than trying to get another FMT treatment potentially. Another thing that some health agencies are requiring is that you need to be able to remove all the bacteria. So if something goes wrong, the idea is that you have a sort of mixture of antibiotics you can give the patient to remove all these bacteria from the synthetic cocktail. But that can be really complicated, especially as you have larger, more complex synthetic communities. And finally, regulatory approval can be difficult to get. To date, from what I'm aware, there's not any synthetic cocktail that's been approved for use. There are a lot of these are in the testing phase. So the challenge will be to get these all through all the safety testing and patients. So this led us to the development of these third generation microbiome therapies. And the idea here is we're moving away from including live bacteria. So instead of saying what bacteria, what are the names of the bacteria that we need to include, we're saying what are these bacteria actually doing and what is their function? And can we just restore that function in the gut instead of having to give these live bacteria? So the advantage of using this technique is that we can really undergo rigorous safety testing because we know exactly what we're giving patients. And it can be administered at a defined dose that we know is effective. So we're not relying on bacteria to colonize and live in the gut and grow to a certain concentration where they can have an active effect. We're actually giving sort of the product of the bacteria. And so we know that it's going to be there at a dose that will be effective. Oh, I sort of just mentioned that. We can give it potentially by a less invasive delivery route. I mentioned earlier with FMT, you have a tube that goes down into your stomach, or you can deliver it from the other end as a colonoscopy or enema. But potentially with small molecules, you can deliver it as a pill or as something you can mix into a drink potentially, depending on how stable your compound is. So there's more options potentially. And again, we're not relying on fecal donations from healthy donors. We're not worrying about whether the healthy donors have antibiotics or something else that might affect their gut microbiota. So it's something that can really consistently produced and be available as well. So one of the early uses of fecal transplants is for a disease called Clostridium difficile infection. It was formerly called Clostridium difficile infection, so but it's just the name's just been updated recently. So for those who may not be as familiar with Clostridium difficile, or C. diff as I'll probably refer to it during this talk, this is a gut pathogen that's often picked up in hospitals, and it's often picked up by people who have undergone antibiotic treatment. So you see here in this diagram, hopefully it's visible to all of you, but we have a person, they say healthy individual, potentially not someone as healthy as anyone walking down the street. This could be someone who's in the hospital as well, but they have more diverse gut microbiota, as we'll say, so more complex gut microbiota. And these patients often get antibiotics. What these antibiotics do is reduce the diversity of their gut microbiota. Once that diversity is reduced, they are exposed to C. difficile in the form of spores. And what I mean by spores is that these are inactive forms of C. difficile, so they're sort of more dormant states that can be easily transmitted and are more stable in the environment to be picked up by other patients, for example. But once you ingest these spores and they get into your gut, if you have this lower diversity gut microbiota following antibiotic treatment, these spores can then undergo a process called germination, where they then start to grow into an active state. And it's actually this active state that causes the disease. So you can see here, it's kind of a gross picture of something called pseudomembranous colitis. But these patients generally have severe diarrhea, abdominal pain, nausea, fever. This is caused by the toxins that C. diff produces, causes lots of inflammation, cell death in the gut, and this pseudomembranous colitis as I've shown here. So antibiotics cause this susceptibility to infection by C. diff, and the first line of treatment is actually more antibiotics. And it can work to treat the disease, though increasingly there's hypervirulent strains and more antibiotic-resistant strains of C. diff. But even if you do have an effective treatment of C. diff with antibiotics, there's always a possibility that these spores are going to remain. And if these spores remain, they can stick around in your gut, and then once antibiotics are stopped, you can undergo a recurrent level of infection because you have still this less diverse gut microbiota, and you can start this cycle of infection all over again. So because we know that reducing the gut microbiota diversity increases susceptibility to infection, this is where the idea of these fecal transplants came, where we're replacing the diversity of the gut microbiota. And actually, it's been seen to be pretty effective. Hopefully you can see here. We can see that we have sort of 4 different groups. The first group is patients that received 1 treatment of FMT. The second group are patients that received 2 treatments of FMT. So in other words, this is sort of adding together the patients that received 1 FMT, a couple of patients failed, and then they added that together to show all the patients that received 1 or more FMTs. This here are from patients that had C. diff infection and just received an antibiotic called vancomycin. And then this is patients that received vancomycin and bowel lavage or bowel treatment. Bowel prep, sorry. So with 1 treatment of FMT, about 81% of patients were actually cured. And this is in contrast to about 31% of patients who received the antibiotic vancomycin and about 23% of patients that received vancomycin and this bowel prep. And there's a few patients that did not get treated with 1 FMT, but when these patients were administered a second FMT from a different donor, this brought the overall success rate to about 94%. So actually, FMT works pretty well for C. diff infection. And actually worked so well that the authors had to stop the study early because it wasn't considered ethical to keep assigned patients to these vancomycin treatment arms. So research in my lab wanted to start looking at why does FMT work to treat C. diff infection? What is it about the gut microbiota and a more diverse microbiota that was able to treat it where antibiotic treatment was failing? And this is where I started to use these models called artificial gut models. You might hear them called hemostats, or sometimes people refer to them as robo-guts. But what these are, I have a little diagram over here. Basically, these are glass vessels that have a growth medium that mimics your gut, and then we inoculate it with a fecal sample. So we're culturing the gut microbiota together as a microbial community, but outside of the host. So we feed it with this growth medium that acts as sort of the food for our gut community, and then we remove culture at a rate that mimics the transit time through the gut. It's heated to body temperature, and we're maintaining the pH, that's the pH of your gut. We're also bubbling through gas to displace any oxygen, because actually your gut is an oxygen-free environment, or an anaerobic environment you might have heard referred to before. So overall, it's mimicking all the different environmental features that you experience within the gut, but actually outside of the host. And I'm just going to show you, and we start to make sure that we don't have any sort of gradient buildup, everything's together. Hopefully I have a video here that will work. This is what it looks like. And just play. So this is just one of the units. And if I zoom in here, this is that glass vessel that's and I showed you. So that's people who've got microbiota growing there. We have a gas control plate pump that we use to feed in the media. That unit controls the pH, controls the stirring. And then this is the growth medium. It's actually, a lot of people think the growth medium is actually the bacteria itself, but no, it just looks like it, but it's not. So usually we have at least 2 of these growing at a time, running at a time, and we can inoculate them with stool from the same donor, and they'll reproduce microbial communities that are very, very similar to each other. So what this does is it allows you to use one vessel as a test vessel and the other vessel as a control vessel. So it's almost like being able to experiment on the same person, but also having the same person as their own control, which you can't do in real life. But some advantages of these models are that they're very tightly controlled. So we're tightly controlling the pH, temperature, speed of the growth medium, and even the composition of the growth medium. So we're going to have less sort of complicating or confounding variables, as you might see in studies of humans, because one of the factors that are really difficult to control in humans, for example, is diet. People eat different things every day, and it's really difficult to say to someone, okay, I want you to participate in my study, but you need to eat the exact same thing every day for so many days. So it's just an easier way to control the composition of the gut microbiota. So if you're giving it some sort of intervention, you know that any changes you're seeing in the gut microbiota are a direct result of your intervention and not actually a result of something else that a human participant could be doing that would affect their gut microbiota. So I was using this artificial gut model to culture the gut and to mimic the conditions found in patients with this C. diff infection. So basically, we took stool from a donor, inoculated into the vessel, we gave them antibiotics and these C. diff spores, and then we let that stabilize. And then what we did was we administered FMT to one of those vessels as a control saline to the other vessel. And that's just the liquid that we use to homogenize the fecal sample to make our FMT preparation. So we have a nice control there. We used a variety of different techniques to analyze our sample, some of the results which I'll mention here today. But we looked basically at the growth of this C. diff, as well as changes in metabolites and changes in the different bacteria from the gut microbiota, so complex microbial communities. We also analyzed fecal samples from patients with C. diff infection who received FMT, as well as the healthy FMT donors, to see how that compared to what we were seeing in the artificial gut model. We performed experiments with pure cultures of C. diff, given different metabolites that we thought might be important to look at direct changes in the growth of C. diff. We also performed a mouse experiment where we had mice that had C. diff infections, and we administered them the metabolite that we were interested in to see how that affected C. diff growth, which I'll get into a bit later. So this plot here is showing you the growth of C. diff over the course of the artificial gut experiment. So beginning, you can see a sharp increase in C. diff, and this is in response to antibiotics that were administered. And then these counts start to stabilize. And then you can see here on day 42, we administered FMT to the vessels in blue, and the saline was administered to the other control vessel. And we saw a significant decrease in the growth of C. difficile in response to the FMT treatment as compared to the saline treatment. So that was looking at the live bacteria, or the actively growing bacteria, but also we also had a chance to look at the spores. So those are the inactive, more dormant states of C. diff. And we can also see a drop in C. diff spores counts as well following FMT treatment. So something else we can do, because we have these artificial gut systems, is we can sample them as much as we want. So we sampled them every other day over the course of our experiment, and we can start to get an idea of what the baseline looks like before we give an intervention. So looking at sort of this top left figure here, you can see sort of the stable baseline, and then in this box region here, it's showing when we were giving antibiotics. So there's a sharp decrease in the growth of these gut microbiota members in response to antibiotics, and then you can see some of these resistant bacteria actually bloomed up. So in other words, they didn't have as much competition as they might have had before antibiotics, so they were able to sort of take advantage and expand within their niche. And then administer FMT, you can see some changes. It's a bit difficult to see in these just because there's so many different colors going on, but one really obvious one is down here where we can see this bacteria that was introduced with FMT treatment. But what we're really interested in is what is happening functionally. So what are the members of the gut microbiota doing, and how are we restoring health when we give FMT to these patients with C. diff infection? Well, one metabolite, we measured a variety of metabolites and looked at different patterns and changes over the course of the experiment. The one that stood out to us is this metabolite called valerate, which is a short chain fatty acid. So that's a group of compounds or a group of metabolites I mentioned earlier in the talk. And we saw some pretty interesting changes. So looking at this figure here, for antibiotics, you see there's some amount of valerate, and the amount of valerate present varies depending on the different donor. So this is a donor in his 40s, this is stool from a donor in his 60s, and this is stool from a donor in his 80s. But when all three donors were given antibiotics, there's a sharp decline in valerate. If we look at follow the lines here in red, we can see these are the vessels that receive saline. So vessels that received saline treatment had no recovery of valerate. It just stayed low for the rest of the experiment. Even after antibiotics were stopped, it did not recover. But when these other communities were given FMT treatment, we can see this sharp recovery in valerate. And this actually correlated with the decrease in the growth of C. diff. Another compound we saw change was this compound called 5-aminovalerate. Sort of shows the opposite pattern of valerate, where we see lower levels pre-antibiotics, but these levels increase with antibiotics. The lines here in red are the artificial gut communities that receive saline, and the ones shown in blue are the artificial gut communities that receive FMT. So it's showing sort of this opposite pattern. Ethanol and propionate were other compounds that we found change as well. Again, sort of showing same patterns as 5-aminovalerate. But what's really interesting here is actually 5-aminovalerate is a compound that can be metabolized by the gut microbiota to produce valerate. Same thing with ethanol and propionate. These compounds can also be metabolized to produce valerate. So what it looks like is that antibiotics have disrupted the gut microbiota in a way that we have inhibited the production of valerate. So potentially valerate was something that might have been inhibiting C. diff growth, but when we're given antibiotics, we lower the levels of valerate, allowing for C. diff to grow. Often get the question, what about other metabolites? There are other metabolites that are often found in the gut, and people often are curious about how they change. So other metabolites didn't show the same sort of clear pattern. We saw, for example, the metabolite butyrate go down with antibiotics, but actually we're able to generally recover. Even in one patient, you see it rebounds to even higher levels than before antibiotics. And with FMT treatment, there was no difference. Same thing with acetate, a slight decrease but gradual recovery without any intervention. And isobutyrate and isovalerate didn't show any obvious patterns. But one metabolite that was very interesting was this metabolite called succinate. So there was interestingly a sharp increase in succinate with antibiotics, but then it actually recovered to baseline levels before FMT was given. But previous studies have shown that succinate is increased with antibiotic treatment, shown in mice. But also it's shown that C. diff can use succinate as a food source. So it's possible that because we killed members of gut microbiota that use up succinate, it allowed C. diff to then come in and use succinate to promote its own growth. So the next thing we wanted to do, after we had the results from our artificial gut model, we're interested in this compound valerate. How is it involved with C. diff? We asked ourselves, well, is this what's happening in people? What do we see in real life humans who receive FMT treatment? So we collected fecal samples from healthy donors, and we also collected fecal samples from C. diff patients both before and after FMT treatment. This is what we saw. We measured the concentration of valerate in their stool, you can see on the y-axis, and we compared it to donor. So you see that validated is present in a donor stool. Those are healthy donors, that's good. In CDI patients before FMT, we can see there's basically no valerate that's been depleted from their gut. But after FMT, we can see that levels of valerate were restored. So that's good. That follows the pattern that we were seeing in our chemostat model, or artificial gut model. The next thing we said, great, but correlation does not always equal causation. So we wanted to show that valerate has a direct effect on the growth of C. difficile. So what these plots here are showing is where we took pure cultures of C. diff and we incubated them with different concentrations of valerate. So you can see here this x-axis are all the different concentrations of valerate, and then this y-axis here is showing you sort of the change in the growth. So we tested 3 different rabbit-types. So there's just slightly different versions of C. difficile, and you see that as you increase the concentration of valerate, you decrease the growth of C. diff in all 3 rabbit-types. And we look at other members of the gut microbiota. There's sort of 3 different scenarios that come up. Either other members of the gut microbiota were unaffected by valerate. Some members of the gut microbiota were actually had their growth promoted in the presence of valerate. And other members only had their growth inhibited by valerate at very high concentrations, so they weren't as sensitive as C. diff. Great. So the next question we said, what happens if we give valerate as a treatment for C. diff infection? And the way that we had to do this was using a mouse model. So we had 2 different groups of mice. First group of mice, well, both mice had C. diff infection. And the first group of mice received PBS as a control, and the second group of mice received a molecule called glycerol trivalerate. So this is administered because valerate, you can see it here, is actually thought to be more quickly absorbed. So they were administered valerate by oral gavage, so in their mouth. And we wanted to make sure that that valerate got to the gut, but there's a possibility it's going to be absorbed into their bloodstream potentially before it reached their gut. So we administer it in this form of glycerol trivalerate, and that's this compound here. So you can see there's a glycerol backbone in the middle, and then 3 valerate molecules attached. And when this is administered to the mice, it actually makes it through their stomach, where it can be then, these valerate molecules can be cleaved off by these intestinal lipases in the gut, and also potentially some produced by bacteria as well. So we measured the amount of C. diff they had in the stool of each of these groups of mice. We found that before we gave them any treatment, the amount of C. diff in the stool was equal in both groups of mice, which is good. And once they received 1 dose of valerate, we saw a slight decrease in C. diff growth. After a second dose of valerate, we saw more decrease in growth. And after that 3rd dose of valerate, we saw the biggest decrease in growth. So there's been a 95% reduction of C. difficile growth after the 3rd dose of valerate, which is pretty good considering patients who receive antibiotics would undergo longer term treatments and might take more than 1 dose per day. Great. So this is some of the previous research I was doing, but some more current research that I'm doing now is looking at the use of these fecal transplants, or FMT, to treat multidrug-resistant organisms, or MDROs. And what I mean by a multidrug-resistant organism, in this case I'm referring specifically to bacteria. These are bacteria that are resistant to multiple antibiotics, multiple classes of antibiotics. And so they're very difficult to treat because just getting more and more antimicrobial resistance, and can lead to further complications. We have to start giving potentially more toxic antibiotics that have more side effects. And there's always this threat of moving to this sort of post-antibiotic era where potentially we might have some classes of bacteria that might not be susceptible to antibiotics anymore, and that's a really scary idea. So we want to try to look at gut microbiota and how do they normally protect us from these pathogens, and can we try to find alternative treatments to gut infections that don't rely on antibiotics. So this figure here is a figure from the World Health Organization. It has a nice summary of sort of different ways that people can develop antibiotic resistance. First being the overprescription of antibiotics. Prescription of antibiotics potentially for people who might have like viral infections, like the cold or the flu, where they're not going to have any impact on your health. Another method would be patients not finishing their antibiotic treatment. Also, the overuse of antibiotics in livestock or fishing, potentially transference of antimicrobial resistant pathogens in hospitals or clinics. Some people might lack the proper hygiene, and also the lack of new antibiotics being developed. So because bacteria have been shown to gain antimicrobial resistance over time, some pharmaceutical companies have sort of been discouraged from spending all the time and money developing new antibiotics, because there's always a possibility that the bacteria can develop resistance and they're going to have to stop. They're going to reduce their sales. So we need to come up with new strategies. So some examples of some antibiotic-resistant bacteria you might have heard of before. There's these carbapenem-resistant Enterobacteriaceae, these extended-spectrum β-lactamase-producing Enterobacteriaceae, vancomycin-resistant Enterococcus, and methicillin-resistant Staphylococcus aureus. Now, how does the gut microbiota fit into all of this? Well, actually, the intestine can be the primary colonization site for certain groups of MDROs. So actually, your gut can serve as a reservoir for these multidrug-resistant bacteria, and these are bacteria that can be responsible for invasive infections, things such as bloodstream infections or recurrent urinary tract infections. So ideally, if we don't have this reservoir of bacteria in our gut, we could prevent infections from developing. So this paper here by Sorbara and colleagues had a really nice example of a patient that developed multidrug-resistant bacteria in their gut and also a bloodstream infection. So this figure down here is just showing you sort of the course of antibiotics that the single patient had gone on in the 10 days leading up to stem cell transplant and the 5 days following. It's on the 6th day that they had developed this E. coli bloodstream infection. So you can see they were on quite a bit of antibiotics, a lot of broad spectrum antibiotics. So we can imagine that would have a big impact on their gut microbiota. And here's what they saw. So this is looking at the composition of the gut microbiota on daily samples. So each of these columns here represents a sample, a fecal sample taken from that patient. And again, 10 days leading up to and following antibiotics. You can see when they were put on this antibiotic here, there was this bloom of this antibiotic-resistant E. coli in their guts. It was on this final day here that they developed this bloodstream infection. And you can see there was really dramatic changes in the composition of their gut microbiota if we compare this day here to this day here. They look vastly different. So as mentioned earlier, there's lots of these group of bacteria, Enterobacteriaceae, that are resistant to antibiotics. So the carbapenem-resistant Enterobacteriaceae or the extended spectrum beta-lactamase-producing Enterobacteriaceae are some I mentioned. And this is what I'm starting off by looking at recently. This figure here is just showing you some data I pulled from a couple of published papers looking at people who are healthy donors, stool donors, and people who went on some antibiotics, some broad spectrum antibiotics. And we can see there's actually quite a dramatic difference in the amount of these Enterobacteriaceae in their gut. So potentially these could be reservoirs for these multidrug-resistant bacteria. So if we look at the healthy participants, you can see there's just a very tiny amount of this Enterobacteriaceae normally present. Sorry. But when we zoom in, looking just at the 0 to 0.5%, we can start to see the levels. So they're very normally present at very, very low amounts. But when we look at our antibiotic treated participants, we can see that they have significantly higher amount of antibiotics, of Enterobacteriaceae in their gut. Quite a broad range. Each of these dots represents a different sample from a different person. And potentially the differences here are due to differences in their underlying gut microbiota, but also potentially differences of the antibiotics these patients have received. So if we look at our artificial gut model, if we take our artificial gut model and we administer antibiotics, we can see there's changes in metabolites. So the lines in blue represent a time series of samples from patients after antibiotic treatment, and the lines shown in pink are the samples taken from the artificial gut model before antibiotic treatment. So you can see the antibiotics cause an increase in the amount of this metabolite 5-aminovalerate, succinate, ethanol, and isobutyrate, and also cause a decrease in valerate, acetate, and butyrate. So that's why researchers have been looking at this idea of fecal microbiota transplantation, FMT, to get rid of these multidrug-resistant bacteria from the gut. There was this study by Julie and colleagues where they looked at giving FMT to patients who had these multidrug-resistant bacteria living in their gut to see how effective it was to decolonize them. So at 1 month post-FMT treatment, 79% of patients were actually decolonised by these antibiotic-resistant bacteria as long as they didn't receive antibiotics within 1 week of receiving their FMT. If we look at patients that did receive antibiotics within 1 week of receiving their FMT, only about 36% had decolonized. But at 6 months, we can see that these numbers increase even more. They didn't do a control group in this case where they had patients that did not receive antibiotic treatment, because some patients on their own can spontaneously decolonize these multidrug-resistant bacteria. But other studies found this smart-based decolonisation rate of about almost 7% in one study and 9% in the other study. So either way, it seems that FMT treatment is having a large impact on decolonizing the gut. But it's not, we still don't understand what the mechanism of FMT is. And it's really not the ideal treatment to be using this group of patients, as these patients are often really immunocompromised and receive a lot of antibiotics. So as you can see here, that's why they receive a lot of antibiotics post-FMT in some patients. It's, again, delivered via invasive administration routes. Mentioned earlier, this idea that we don't know the long-term effects of FMT. And again, we don't know who is the ideal donor, as not all FMT donors are created equally. So really, it'd be more ideal in this group of patients to develop a therapy for decolonizing these multidrug-resistant bacteria from the intestine before the patients go on to develop invasive disease. So this is very new work from my lab. So I just have some preliminary data to show here. What we had was our artificial gut system, and it was inoculated with stool from a patient that had intestinal colonization with not one, but actually 2 multidrug-resistant bacteria, carbapenem-resistant E. coli and this carbapenem-resistant Klebsiella strain. And in this first plot here, we see that the lighter pink is the artificial gut model that received saline, and the sort of red color is the artificial gut model that received FMT. So FMT treatment actually caused a 99% reduction in E. coli, multidrug-resistant E. coli. And we look at this other multidrug-resistant bacteria, Klebsiella, we can see a 95% reduction with FMT treatment. So it does seem that FMT treatment is working, and our next steps are really going to be to identify, are there any specific metabolites or enzymes that are produced by the gut microbiota that are part of that FMT mixture that are responsible for inhibiting the growth of these multidrug-resistant pathogens, and can we try to come up with this third-generation therapeutic where we have a specific metabolite or enzyme or safe to give these patients. And you can even take it while they're in the hospital where they might be receiving a lot of antibiotics. So really, our goal is to develop a new method for the decolonization where we're reducing the risk of patients developing these invasive diseases, reducing the recurrence of invasive diseases in chronically colonized patients, for example, recurrent urinary tract infections, and we're reducing the spread of these multidrug-resistant bacteria to susceptible patients. It gets spread in settings such as hospitals or care homes. So that's just an example of some bacteria that can have their growth affected by the gut microbiome. But this idea of colonization resistance and restoring colonization resistance has a potential to impact other gut pathogens as well. And there's been some cases reported for all these pathogens I've shown here. So it really just has potential to be developed further as new treatments, but we're moving away from antibiotics and just trying to restore the functions that we're seeing in healthy guts to protect us from these infections. So that's where I'm going to end it for today. I just want to take a second to thank my collaborators, Ben Mullish, Roman Ghani, Julie McDonald, who helped with this study and for the work with multi-drug-resistant bacteria, and as well as the funders. And I guess we'll start taking some questions. So if you have any questions, just post them in the Q&A. I think I saw some going up earlier. Great. So first question here, I'll just read it. Could you remove bacteria if something went wrong by using some sort of suicide gene? That's a good question. I don't think that's something that people have been looking at. I think one of the complicating factors with doing that would be we would then be genetically modifying the bacteria. And as soon as you start talking about giving genetically modified organisms to humans, that's where it starts to get a lot more complicated. So I mean, there's always a potential, but it might make sort of the more regulatory process behind it more complicated. Whereas if you're giving a bacteria that's in the gut found naturally, it might be easier to get that approved. So potentially in the future, but right now, I think it would be maybe more challenging. Could Clostridioides be described as an opportunistic pathogen? Yes, I suppose you could think of it that way, because if you, it is taking advantage of the fact that you have sort of this antibiotic susceptible, your treatment makes you more susceptible. So if you're a healthy person, you haven't had antibiotics for a while, you have a healthy diet, you think you have a diverse microbiota, you're going to be generally fine if you were to accidentally ingest these spores. But it's really when you are receiving antibiotics that you start to have the growth and the toxin production by the pathogen. Next question. Why was saline used as control rather than distilled water? Good question. The main reason for using saline is it's a liquid that will be sort of stabilizing for the bacteria. So if we use just purified water, the bacteria have osmotic effects and the bacteria might burst, versus saline, it's sort of a isotolerant compound where it's just trying to maintain the viability or the bacteria from the gut alive more stably than water would. Okay, next question. How long after the end of antibiotics exposure are the levels of Enterobacteriaceae high? They're linked with. So I think that's a good question. So in humans, I think some of these can be difficult to perform because longitudinal studies in humans are challenging in that they can be expensive. You have humans that are eating different diets. I don't think I've come across a study that has looked specifically at for how long it takes for the gut microbiota to recover. I think it really probably varies depending on the antibiotic that you've taken. One study I read a little while ago was looking at an antibiotic called clindamycin and looking at sort of just overall changes in the gut microbiota, not Enterobacteriaceae specifically, but sort of overall how did it affect the gut microbiota. And in some cases, they were reporting even 2 years later, there was still changes, and that was the end of their study, and they had not recovered to that initial level. So it really depends. You might have some bacteria that recover, some bacteria that might never recover. And it's not to say that that might be a worse microbiota than before antibiotics, because this is coming into the idea of sort of composition of gut microbiota versus functionality, because your gut microbiota does have this level of redundancy where different bacteria can perform the same function. So losing a bacteria might not necessarily be a bad thing if you have another bacteria that can perform that same function. Okay. So what analytical technique is used to determine the concentration of various metabolites? Would there be a benefit to direct measurements in the bioreactor as opposed to drawing out samples for analysis? So there was a couple different techniques we used. The valerate compounds and some of the other ones I showed today is a method called NMR. NMR, it works pretty well by drawing out a sample, and you can put it into the freezer and then take all the samples out at once and run them all. There is talks of having a method of NMR where you could potentially do a live analysis where you might be able to pump a sample out, run it through the machine, and get live data. That might be good if you are looking for immediate effects where you need to know immediately what's changing and maybe make some sort of change in response. But it's very complicated to do in anaerobic conditions, and there's not very many places that can do it sort of specialized and more of a new developing idea. So if in this case we didn't need to know live what was happening, we did have the changes in the plate counts of C. diff, for example, to know that we were seeing changes in the growth, and that was some information that we were feeding back. So in that case, it was not necessary. But we also looked at changes in bile acids. Which I didn't show the data here just for time's sake, but if you're interested, it's published out there as well. But changes in bile acids are known to have effects on C. diff growth as well by inhibiting vegetative growth, so that live bacteria, and also inhibiting spores. And we also look at changes in the gut microbiota, so that would be using DNA sequencing, 16S rRNA gene sequencing. Okay. Sorry, I'm just scanning some questions here. Okay, so he collaborated with anyone at Imperial who is modeling the equivalent small molecules that mimic bacterial interactions for third generation FMT, or developing the small molecules. So that's some work that I'm doing in my lab now, is looking at these third generation FMT ideas. But my collaborators here, Ben Mullish, Ramagani, Julie McDonald, and Mark Theres, we're all working on in this area together. So yeah, but there are other groups around the world that are looking at this as well. There are a lot of really great studies out there too, but it is a generally new area. Okay, so would you say that there are factors that would determine whether the first or third generation therapeutics are more suitable for patients? Yeah, yes, I would think that, as I mentioned earlier, immunocompromised patients are more of a challenge with live bacteria because their immune system is compromised. If you unknowingly transfer something that may cause disease in an immunocompromised person, you might not know you're doing it because a healthy donor has an intact immune system, so they don't have any sort of signs of disease or development of disease. But if you transfer that to a patient who's immune compromised, it's always a potential. That's where I talked about these antibiotic-resistant bacteria earlier, where there were some patients that died, and that's because they had a bloodstream infection develop after the FMT. So in that case, I think FMT is probably not ideal to give those patients. Second generation is going to be more safe, but we still need to be very careful about what you're giving them in these synthetic cocktails. I've not seen any issues with synthetic communities published so far, but I know I've seen some papers mention probiotics in intensive care unit patients. You can sometimes see some of these probiotics showing up in the bloodstream of these patients. Not described very often, but it's not impossible. So perhaps in those patients, you really would be preferring to give them something like a metabolite or enzyme. It's not a live bacteria. You stop it, it washes out of their system. Nothing's going to colonize within them. It might just be a bit safer. Next question. Are there risks of affecting neurology through FMT due to the links between the gut microbiome and psychiatric disorders? That's a good question. I think that's sort of a newly developed area, looking at the link between the gut microbiome and these disorders. So I think there's been a lot of studies just looking at changes in the gut microbiome in different groups of patients, but there hasn't been as many studies sort of investigating or demonstrating the direct cause and effect. So I can't say for sure what the effects will be of an FMT. I think the idea is that if you're giving a healthy gut microbiota, hopefully you're not, promoting harm in any way. But I think that's why it's really important that we have a clear understanding of the long-term effects of FMT, just to really make sure that it's safe. So I guess I just say we're not there yet. We don't know. How important is temperature? So I assume this is in regards to the artificial gut models. So and temperature is pretty important. So if we're looking at culturing bacteria from the gut, we want them to be growing sort of under the conditions that mimic the human gut as closely as possible. So 37 degrees is temperature that we use. If we start to raise that or lower that, you're going to start to not encourage the growth of these bacteria. So some bacteria might grow slower at colder temperatures or not at all, and some bacteria might be killed at higher temperatures because you start to denature proteins if you start to get really high. Okay. We are got time for maybe one more question. We'll see how we go. Okay, so is there a high variability of bacterial and metabolite composition between donors, even between donors whose samples could be efficacious? Does this not make standard treatment extremely difficult? Yes. So people's gut microbiota compositions and the metabolites they produce are variable between people, and they're actually even variable between the same person over time. So I had fecal samples collected from the healthy donor, taken one day apart, and just by looking at the fecal sample by eye, you can see differences. And if you analyze the fecal sample, you can see differences as well. And that, I think, is really down to the diet. So they had very different meal consumed one day, and the next day they consumed something else that could affect the composition of their gut microbiome. And so sampling the same person day to day, week to week, month to month, it's going to be generally stable, but you can't guarantee it's going to be the exact same. So that does make it challenging, and especially when you have, you have a metabolite that you want that fecal sample to contain, it can be challenging as well. So I think that's why if we can identify exactly what metabolite or enzyme is being produced, we can more tightly control what the patient's going to be receiving. Because even if this fecal sample is the exact same, and you're giving it to two different patients, what bacteria they have in their guts remaining, because if you take antibiotics, you're not completely sterilized, you still have some bacteria present in your gut. So depending on what bacteria are in the patient's gut before they receive FMT, what antibiotics they could be taking before and after the antibiotics can affect the composition, even what diet they're taking, it's all going to affect what bacteria from the FMT are going to be able to live in the gut. So it's really difficult to try to predict when you're giving an FMT what bacteria are actually going to live there. Let's see. One more question. Let's see. So could there be a way to reduce the potential harmful effects of bacteria in the microbiome so that FMT can be used in immunocompromised patients? Yeah, so I mean, I think the ideal thing here is to move away, ideally, from using live bacteria if we can, to using enzymes, metabolites, or if we're going to be giving bacteria, there should be bacteria that we know are not going to be able to cause disease, but also bacteria that we know are going to be effective at decolonizing these antibiotic-resistant bacteria. So yeah, that's the challenge. I think we're going to just stop there because we're running out